Hello everyone and welcome to this audio this audio wherein there will be an overview on this particular this research poster presentation for the International Paediatric Transplant Association, or IPTA 2023. This research po- research will this research poster presentation looks at transplant immunobiology, and in context of a chronic kidney disease review. This was a collaborative effort between different academic institutions, and also wherein the Renal Patient Support Group, the RPSG, had a sort of a basis of involvement as well. So what I thought I'd do here is provide some context in around this research poster presentation, and so in and around the background, the immune system protects the body from infections by pathogenic organisms, and it is composed of a complex constellation of cells, organs, tissues arranged in an elaborate and dynamic communications network. The immune system is, in its simplest form, a cascade of detection and adaptation, culminating in a system that is remarkably effective. Leukocyte and lymphocyte migration and trafficking from peripheral blood into affected tissues is an essential component of the inflammatory reaction to virtually all forms of injury, and is an important factor in the development of many diseases. Advances in the past few years have highlighted the central role of a family of chemotactic cytokines called chemokines in this process. Cellular adhesion molecules also have also been important in allograft or tissue rejection, as these molecules allow protein receptor binding. Chemokines also help to control the selective migration and activation of inflammatory cells into the injured tissue or allograft or graft. Chemokines and corresponding receptor ligands migrate during inflammation, and there is some evidence that certain negative states, such as stress, depression, and/or anxiety, can directly affect physical immunity through production of stress hormones, such as, for example, catecholamines and glucocorticoids. And there is some indication that negative states may lead to faster progression in certain diseases. So, for example, in human immunodeficiency virus, or HIV, and other conditions, through direct biological mechanisms. Negative states may also indirectly affect disease processes through influence on health behaviors. So, for example, depression has been related to many risk factors for poor health, and stress with regard to Hans-Selye's general adaptation syndrome in 1936 was and has been considered with commencing various disease aspects, and looking and also states. So healthcare also incorporates another sort of model called another model that has been referred to as the biopsychosocial model. The biopsychosocial model is also a sort of a technical terminology for the popular concept that links the mind and body connection. Now, the biopsychosocial model, was developed by a psychiatrist, George Engel, at the University of Rochester, and works was initially published in 1977 in and around an article of Science. And the biopsychosocial model draws a distinction between the actual pathological processes that cause disease, and a patient's perception of health, and its and the effects on it called or referred to as the illness. There are also theories that directly affect the immune system, and there are investigations that show this to be the case. And in synchronicity, the in synchronicity, in context of the mind-body connection, as well, it has been referred to as neuroimmunology, and thus looking at the sort of psychology, the neuro the neuro 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 the neuroendocrino no, neuro and neuroendocrinology, and also immunology or immunobiology. And so the aims and objectives around this works was thus: so looking at the entities based on and in around chronic kidney disease, transplant immunobiology, and neuroendocrinology. The aims and objectives was to look at these entities, as a basis for this sort of this sort of review. And so, in and around transplant immunobiology, and in context of a retrospective, there have been several investigations exploring clinical and experimental observations that indicate that immune responses may be modulated by the central nervous system, or the CNS. It was works by Neveu and Limaul, in the 90s, and in 1990, that highlighted that the immune system and the central nervous system are known to communicate through the endocrine and autonomic nervous systems, and also inform that complete regulatory loop between immune and neuroendocrine systems exist. It was Wagent and team in the 1990, who also informed that there is a molecular basis, and there could be a molecular basis for a bidirectional communication between the immune and neuroendocrine systems, and the recognition of stimuli is converted into the formation is converted into information in the form of lymphokines or lymphokines, monokines, and hormones, and a message received by the neuroendocrine tissues. And it was and the nervous system recognition of stimuli can also be converted into chemical signals that can be relayed into the immune cells, resulting in physiological changes. It was Bedofsky and Delray in 1991, who informed that interactions imply the existence of afferent messengers derived from the immune system, such as lymphokines and monokines, capable of integrating immune neuroendocrine circuits. At the time of peak at the time of the peak immune response to innocuous agents antigens, as well as following inoculation of infective uh, eh, eh, agents, the endotoxins, or tumor cells, immunological cells release cytokines which can stimulate the pituitary adrenal axis. And so the acute effect of these cytokines is mediated by stimulation of the release of corticotropin, a releasing factor from the hypothalamus, um, of the hypothalamus. And Grochowitz and team in 1991 that highlight that conditioned immunosuppression or implementing investigation using investigation in around a taste aversion paradigm had been demonstrated in several laboratory investigations, and the team reported that survival of cardiac allografts in rat models can be or could be prolonged by immunosuppression using a particular pharmacotherapy referred to as cyclosporin A as a stimulus in a taste aversion conditioning. And conditioned animals received saccharin as the conditioned stimulus paired with the injection of cyclosporin A at 10 and 6 days at 10 and 6 days before transplantation. Early investigations suggest that behaviorally conditioned immunosuppression may have important clinical implications as an adjunct to drug treatments in and around transplant immunobiology. It was Grotzel and Sridharan in 1992 who informed that integration between several immunobiological functions in host defense may reveal novel mechanisms of cellular communication and biological adaptation. It was works by Ottaway and Husband in 1992 highlight that many of the alterations in immunity resulting from central nervous system from the CNS activity can be explained in terms of changes in in a in a and through a feedback loop. It was Klein it was Klein and team in '93 that highlight cellular molecular basis for the modulation of sorry, my apologies. There is uh, it was it was it was it was it was It was Grotzo and Sridharan in 1992 informed that integration between several uh, immunological uh, immunological functions in a host defense may reveal novel mechanisms of several communication and biological adaptation um, and biological adaptation. And Ottaway and Husband in 1992 highlight that many of the alterations in immunity resulting from central nervous system activity can be explained in terms of changes uh, in uh, in lymphocyte migration in response to endocrine signals, neural signals, and neurotransmitter release, and the direct and or direct contacts between nervous and immune system. It was Kropinig's work in 1993, where the research looked at and suggested that the immune system is active even in a bidirectional feedback loop. And it was Klein in 1993 that highlights cellular molecular bases for the modulation of host resistance is not fully understood, but involves altered functioning of both T lymphocytes and cells of the hypothalamic pituitary adrenal axis. It was also Klein in 1993 also looked to highlight that there is a production of cytokines and hormones produced by the immune system and the brain. It was Wagen and Blalock in 1995 that highlight that prospective investigation should seek to investigate the physiological role that cytokines and neuroendocrine hormones between immunology and neuroendocrinology. It was Perner and team in 1997 informed that in terms of the endocrine and immune responses, acute high intensity physical activity and stress may be considered as a subcategory of stressful active coping. And neuroendocrine responses include increases in catecholamines as well as elevations in cortisol under high but relatively low sympathetic activation. And immune system responses include increases in natural killer cell number uh, and cytotoxicity and suppressor cytotoxic lymphocytes. Chronic life stressors influence acute sort of responsive in context of cardiovascular, endocrine, and immune responses to acute stresses. And physical activity also causes an increase in beta endorphin. And because of the beta endorphin because of the beta endorphin, the important immune units and parameters generally stay blunted. And in the pre-transplant phase and during sort of stressful periods, there is no beta endorphin. And so the immune components such as chemokine receptors, CD8 T cells, natural killer cells, may influence and propagate or prompt acute allograft rejection post transplant. It was works by Ferenczek and Stevanova in 1997 that informed that neuroendocrine system influences and or modulate the immune components that link to physical stress. It was Danza in 1997 that informed that there is evidence that different perceptions of stress have different physiological consequences, and different ways of coping with stress result in different consequences on immunity, the nature of outcome of which depend on the type of immune response. It was Webster in 1997 that demonstrated that glucocorticoids and catecholamines favor T helper cells type 2 or Th2 over T helper cells Th1 or over Th1 type immune cells and mediators by controlling the production of specific regulatory cytokines. It was Gaillard in 1998 that informed the immune neuroendocrine interactions are involved in numerous pathophysiological conditions and seem to play an important role to maintain homeostasis. The mutual information circuit inside of the immune, nervous, and endocrine system suggests that there is a link with pivotal regulatory role in living organisms. And Ferencek in 1998 assert that there is the immune system that the immune system can be referred to as a sixth sense, and may ultimately provide a sort of novel understanding of physiology required for disease and stress involving immune neuroendocrine communication. It was Klapheek in 2000 that informed that though there are questions that remain sort of unanswered, it appears reasonable to include that the impact of depression and possibly stress amongst factors that may affect the use of immunosuppression in transplanted recipients. It was work by Hennig and team in 2000 that conduct a study on the mechanisms of changes in lymphocyte numbers after stress. And within the research strategy, this team identified that catecholamine is linked to stress and found it is of major importance surrounding frequency what had been referred in their investigation as a mild leukocytosis, an increase of white cells of particular or white cells in context of an immunological basis. And in another investigation, Hennig, Hennig Hennig and team in 2001 demonstrated that cortisol seems to play an important role in stress-induced redistribution patterns of CD8 cytotoxic T cells. And this, however, can be explained by different migration processes between cells. So, for example, different cells of migration, and therefore different glucocorticoids influences on target tissues. In works by Gaillard in 2001 informed that the crosstalk between the immune and endocrine system is important to homeostasis, since the interactions can produce various appropriate adaptive responses when homeostasis is threatened. It was works by Harbers in 2003 that highlight that in the field of psychoneuroimmunology, or PNI, PNI hormones, peptides, and transmitters, neurotransmitters regulate the immune system. However, understanding the complex interaction of systems is still a challenge for the 21st century, and pathogens, tumors, and allergens are detected with greater sensitivity and specificity by the immune system. However, George Engel's biopsychosocial paradigm is perhaps one of the main models when dealing with most human diseases and disorders. The biopsychosocial model also encourages reading into areas not traditionally associated with transplant immunobiology. It was Blalock in 2007 who also informed that as a sixth sense, the immune system is a means to signal and mobilize the body to respond to these types of challenges. And investigators sort of prospectively should acknowledge that research surrounding transplantation should not be restricted to single organs, and that the psychoneuroimmunology aspect may have a role in prospective investigations, and this may produce better understanding of the immune system, and it may be a means of attaining T cell tolerance post transplant. This research poster highlights a basis also around the principle of chemokine action. So, for example, chemokines released by stimulated cells act locally in context of immunology. Chemokines are produced by intrinsic cells as well as by infiltrating cells. Chemokines act through G protein coupled as seven transmembrane receptor induce a sort of a calcium influx. Chemokine receptor expression is cell specific. Chemokines bind to proteoglycans and extracellular matrix, and chemokines induce hepatotaxis. So, for example, cell migration along surface gradients. Chemokines can activate adhesion molecules, and chemokines can induce oxygen burst and the release of granules and proteinases by leukocytes. And chemokines control aspects of immunomodulation, angiogenesis, and hematopoiesis and development. The works around the principles of actions have can be identified through the works by Segreger and team from 2000. And in context of sort of prospective investigations, it was works by Al-Arabi and 2000 in 19 and team in 1997 that assert that pre-transplantation plasmapheresis together with immunosuppressive drugs, so for example cyclophosphamide and prednisolone, can be helpful in the removal of human leukocyte antigens, or HLA, in immunized patients awaiting renal transplantation, for example, and it can be considered a valuable approach to increase the probability of successful transplantation in one investigation by Nafar and team in 2006. There is suggestion that plasmapheresis, with or without other therapeutic measures, may have a role in the salvage of allografts with early acute dysfunction, where recipients may be resistant to conventional therapy, and the team conclude that allograft prognoses in recipients with acute humoral response who respond to plasmapheresis, for example, can be comparable to that in other renal transplanted recipients. In vitro investigations using interleukin 15, interleukin 2, and interleukin 15 receptin alpha stress cellular adhesion molecules and relevant monoclonal antibodies should be could be investigated with chemokine receptors such as CCR5, CCR7, and CXCR3 in a T cell culture system to see if this affects T cell migration post transplant. And using peripheral blood mononuclear cells PBMCs would also be advantageous. And in around the discussion here, whilst there have been some collaborative approaches between discipline and practices which incorporate laboratory investigations, understanding chronic kidney disease or CKD in context of transplant immunobiology is pertinent, and indeed stress adaptation and mechanism aligned to psychoneuroimmunology or PNI are topics to contemplate across solid organ transplant specialties. And in context of conclusion, again, using chemotaxis assays with flow cytometry would aid to give graphical data and linking specific endocrines such as cortisol, adrenocorticotropic hormone or ACTH, and glucocorticoid hormone with the above in context of implementation of chemotaxis assays could be advantageous. And stress may have a specific effects with regards to T cell migration post transplant, and so instigating migration investigations or studies is one of the most useful ways to look at patterns or look at cell patterns. And without laboratory investigations and collaborations between transplant immunobiology and endocrinology in context of chronic kidney disease, it is a challenge to state any specific correlations. In this research poster, there is a sort of a list of references that listeners can delve into. The research poster is accessible through the ResearchGate page, so listeners are welcome to go onto the RPSG ResearchGate page and access the research poster that way. Wherein the research poster again has a list of references as well. And so this concludes this audio in and around transplant immunobiology and a chronic kidney disease review. This was a research poster overview in context of works that have been put forward for the International Paediatric Transplant Association or IPTA 2023. Thank you for taking the time to listen in.